Yeah, good morning, all of you. A warm welcome to join this FDP. The today's talk, what I will discuss is 3D bioprinting. Okay, so that this is also my research area, and I'm working, as already mentioned, I'm working as an associate professor in the biomedical engineering department at IIT Hyderabad. So in IIT Hyderabad, like in biomedical engineering department, we have all the programs, like starting from BTech in biomedical engineering. That we are the first IITs to start BTech in biomedical engineering. Then also we have MTech in biomedical engineering and also medical device innovation program. And also we have PhD in biomedical engineering. And we are like almost 10 faculty working on diverse fields. Okay, and I hope many of the participants here also you are from different fields. So what I'll do, I'll discuss this from a very beginner's perspective. Okay, so the 3D bioprinting, because it's the advanced field of research, but I will start from the very basic, so that if you and also if you have any questions, I'll be happy to answer. Probably we have some time for question answer at the end. Okay, so let's start today's thing. 3D bioprinting, most of the thing, this is the content outline of the talk today, so I'll follow this pattern. So 3D bioprinting, many of you are aware of 3D printing, right? 3D printing is nothing but when we fabricate any three-dimensional structures by a layer-by-layer deposition of or layer-by-layer additive manufacturing process. So that's called 3D printing. So where we start with the virtual blueprint like a CAD model, and then the CAD model is used for development of this 3D structure. 3D bioprinting, it is little different from that, where exactly here we use the main purpose of this technique is to develop artificial tissues and organs. So you know very well, our body is made up of cells and some extracellular matrix components, because cells and the cell and basically the housing of the cells, right? Those are materials. So they definitely, then when we go for bioprinting, we need these to make these two raw materials, right? Cells and biological relevant materials. That is, that material can go into the body, and then that helps in tissue regeneration, aids in regenerative process. So that's why for a 3D bioprinting process, we use cells plus matrix materials, and those that basically many of the times those are mixed together, and then that is used for as a bioink for fabrication of this 3D structure. In certain cases, we also only the materials are printed, and then on top of that the cells are deposited or cells are seeded. So there are various types of bioprinting approaches, but this is the whole, in a nutshell, this is the bioprinting approach, where in a layer by layer manner we print the cells plus matrix materials, and then we come up with a functional tissue construct. The process, the general or the typical 3D printing process starts with the CAD modeling. There we develop the solid model using any CAD software. But in case of bioprinting, because our interest is to print tissue or organs, so we don't start all the time with CAD. Making the CAD models by ourselves. What we start with, we generally take help of the various bioimaging modalities like CT, MRI, all this. Okay, and then we get the three-dimensional data, that 3D image of that particular tissue construct. Suppose in this case, just a minute. Suppose in this case, the a CT data is being used. So now you know that the 3D image that is available in the form of DICOM, that we can convert into STL format, so that our printer can understand the STL format, so that can be used as a CAD model that can be used for printing the 3D structures. Now, when you are done with the CAD modeling thing from the CT data or MRI data, then that is being fed to the 3D printer, or that is loaded to the 3D printer, and there using the printer command, then we can print the structure. Now, to print a 3D tissue or organ, we need various other things, like we need cells, we need biomacromolecules, we need hydrogel, and some scaffolding material. So all these things are required. All these things are processed and prepared in advance before the 3D printing. Then the actual 3D printing process is done. Now that part can go for actually, and at this stage we call them pre-tissue, because these are not matured yet. But then when then further these are matured in a bioreactor, and then that can be used for implantation purpose. This also can be used for other purposes. I will come to that in a moment. So this is a video that I have collected from some other sources. The source is already given in this thing. I hope you can able to hear the sound of this video. Like here, what is that? This is the bioprinting head, where this bioprinting head is being. Now here, these are the different nozzles where we can actually load different biowings. In different biowings can be loaded in different printers. Suppose we want to print 3 different cells, so that can be differently done. Now here they will show you how different types of cells can be allowed. Suppose the first nozzle here, endothelial cells, one type of cells are loaded. Second, muscle cells are loaded. In the third one, stem cells are loaded, and the fourth one probably some other materials or stem cells are loaded also. So basically 4 different types of biowings can be used for printing the structure. Suppose other inks can be used for other materials, like suppose other biomaterials. So when then simultaneously one by one we can by using this extrusion based printing method, we can print the tissue structures. So yeah, we can print the we can pattern the cells in a basically we can pattern them in a particular fashion, in a layer by so depending upon the geometry that we want to create, we can create any kind of pattern. And then suppose in between, in the middle, we want to put something like some materials to fill the gap, so that can also be done. So this process is repeated over and over again, so to make the whole 3D structure, to get the geometry of a particular tissue. Now here they have shown you a circular pattern, but you can actually basically you can create any geometry, any type of structures, any 3D structures you can create depending upon the target organ. That can be done. So this is how the bioprinting is done for the artificial tissue or the organ constructs. Now, soon after printing, then we allow it to mature, and then further culturing further to develop for the cell viability, cell proliferation, all these things. I have collected this video from the YouTube. This is the source. You can also see there are many videos available on the YouTube. The next thing I'm going to talk about, what is the purpose of this 3D bioprinting? Because now you see there is a huge interest in 3D bioprinting. Now many people, they work on, can we print this structure, can we print this tissue, can we print the other tissue? So it is true that this 3D printing has got huge prospect and huge applications in biomedical area. Now here, the very first thing we can do with 3D bioprinting is actually we can print different artificial tissues or organs that can be used for tissue engineering purpose. Tissue engineering is nothing but where you use different engineering principles to develop a artificial tissue, and that can help to regenerate a particular damaged or diseased organ. Now with the bioprinting, we can develop like bioprinted skin, cartilage, vasculature, liver nodules. All these different things can be done. Now the next thing we can do with 3D bioprinting actually, because this process, you know that this process starting basically from scratch, like we are developing the tissue from the scratch, nothing is there, and slowly by depositing material, by depositing cells, we are developing that artificial tissues construct, so that in this process, so we can understand the tissue development, meaning because how the tissue is developing because we are printing, during printing they are not just a cluster of cells and materials, but when they mature, they become a functional tissue. So that we can understand by this. So human physiology, pathophysiology, that can be understood by this. Also this can be also used for different in vitro drug screening purposes, like probably you heard of human on a chip, lab on a chip, liver on a chip, lung on a chip, these kind of things. So we can develop actually various tissues or organs, and that can be, if we can, that further can be connected by some microfluidic chip and then that can be systemic response can be obtained from that. So there are various applications of this thing, and that's why we use 3D bioprinting for different applications. Now depending upon the scope of this, or depending upon the I should say depending on the mechanism or the operation, the 3D bioprinting can be categorized into 3 different classes, like inkjet bioprinting, extrusion bioprinting, and laser assisted bioprinting. In inkjet bioprinting, there it is very similar to your office printer. Many of you have in your offices at home, the inkjet printer that works on a principle like the developing generating droplets, right? So these droplets are basically sprayed on a piece of paper, and depending upon your pattern, like when you take a printout of some object, some image or some document, then depending upon the pattern, how the letters are arranged, how the what, and also in the image how the grayscale is arranged, or suppose in case of color image also how the intensity vary, depending upon that basically the droplets are created of that particular color or maybe in black color, and then that is being printed. Now it was for bioprinting, when we want to use that, then we create droplets. So these droplets again can be patterned, the three-dimensional pattern can be possible, and there many a times we use some support material, because with droplets we can make a 3D structures. Sorry, we use some kind of biopaper. This biopaper is made up of, mostly this biopaper is made up of some collagen material, and then we can make the whole 3D structure. Right now, then we can put one layer after another simultaneously. These layers can be printed, and then when the whole thing is done, then we can allow them to mature further. By in this process also, what will happen, the biopaper will degrade, and we can get very good three-dimensional structures. So now this is the final structure that we can get. Suppose a cylindrical structure, you can think suppose like this can be your food pipe, or it can be a blood vessels, or it can be a trachea, right? So these kind of things can be generated with this 3D bioprinting approach. Here the bioprinting works actually basically here two types of mechanisms are there. One thing is where a heater is connected to the capillary of the printed. When the heater is heated up, a bubble is produced, because of this the media, printing media is mostly aqueous, so the bubble is produced, and that bubble can dislodge some material from the below, and then so that it can be the droplets can be generated. In case of piezoelectric, there are other mechanisms where the piezoelectric crystals are attached. Now with this piezoelectric crystal, you know that if we give electrical energy to this piezoelectric crystal, it converts into mechanical, so that there will be a vibration upon giving some electrical signals. So this vibration creates a disturbance in the liquid column, and then by that the droplets can be generated. So depending upon any mechanism, the main purpose is to generate droplets. So now when these droplets are here, this is the schematic of the same thing. Now depending on the droplets, creating droplets, these droplets can be printed. Now here, this is a 3D by inkjet bioprinter. This looks very similar to our office printer. There are a few differences, like the paper tray has been replaced with a plate, or any petri plate, any tissue culture plate can be used for printing the tissue in this plate. And the cartridge, what is the cartridge? We know that we generally buy this inkjet printer cartridge. Now this cartridge is replaced with a Biowing. The Biowing is placed inside the cartridge, so that can be used for generating droplets. I have a video. This is also I've collected from the YouTube, so you can see here how this bioprinting process is done. Just a minute.
Achieving cell printing requires some clever bioink for.
Yeah.
Using inkjet printing, we can print living cells with extremely high resolution, even one cell at a time. Achieving cell printing requires some clever bioink formulations. If normal cell culture medium is used as the ink, the living cells can burst due to the high stresses of the jetting process. But when biopolymers are added, the cells can be protected and printed successfully. Printing of living cells can allow for development of novel wound healing strategies. Living cells derived from the patient could be deposited at the site of injury. Different types of cells could be built up in sequential layers. Such a strategy could accelerate healing or reduce trauma after surgery.
Okay, so this is in nutshell. This is the inkjet 3D printing, where cells are used to develop the Biowing. Basically, cells are suspended within any kind of buffer or media. Then that is being used to 3D print the structure with this. So generating droplets. So the droplets containing cells that will be deposited in a particular pattern, and then we can use it for developing the tissue structure. So here you can actually basically use any types of cells, and that can be done. The next printing technology is extrusion-based bioprinting. Here, this was developed in 2000 by this company, Envision Tech. Now, here the technique or the mechanism is very simple. We have some syringe here. So we said this is very similar to our injection syringe, and where we load the material. Like this is the hydrogel loaded cells. that is Mr. Cells encapsulated hydrogel that is loaded here, and then we can push that by pneumatic or by piston driven pressure to extrude the material, and that can be deposited on a collected plate, and so in a layer by layer manner we can do that. So this is one such bioprinter, Envision-Tech 3D bioplotter. We have this in our laboratory. This is the printing from our lab video. Here you can see that the bioprinting is actually basically printing line by line, layer by layer. It is being printed. And here this is printing of gelatin. We can load soil cells with this material, and then the whole printing can be done. Right? This layer by layer manner, we can manufacture the whole 3D structure. So here this is printed a crisscross pattern where a simple crisscross 090 pattern is created, but we can create any kind of structures within this printer. Right, so this has various components like this, starting from the like some components are there for the software for generating this printing path, printing digitals, all these things. There can be some deposition mechanisms, and also some injection mechanisms. So all these things, various components are there. And there are some process for all these different 3D bioprinting. They have some process parameters, and you know very well here, like viscosity, nozzle diameter, nozzle type, pneumatic pressure or mechanical force, extrusion rate, print speed. All these things are the different process parameters. So now this is a video here clearly showing how this actually 3D bioprinting is done. We did this extrusion-based bioprinting process. You see here, this is the material. They have loaded some dye within the material to actually visualize them. Now every layer, different dyes are being used. You see the layer by layer, it is being created, this structure is created. The second layer is done. Now the third layer is going on. When the third layer is again, so they have printed the on top of this thing. And if you notice, they are creating some kind of porous structure, right? I'll come to that. The porous structure is actually important because in many of the tissue construct, the cells will be loaded inside the structure, right? Inside this line, right? Now where the cells are loaded inside this line, the cells need oxygen, nutrition for their survival. And this structure this will be cultured in a media. Now if it is there, the porosity is there, then the media can perfuse or can diffuse through this porous structure, and then the cells present here, they can get the nutrition or oxygen by diffusion. So that's why this porous structure is get. If the porous structure is not there, because the media can reach to the structure, reach inside the structure to a certain extent, then it will not be able to reach, because the diffusion cannot work for very deep structure. So that's why the porous structure is mostly created for this. Now the extrusion-based bioprinting strategies can also be of different types. Like when we print only hydrogel, cells laden hydrogel, then that is one type of thing. But many times our structure is so complex then that we need some support structure. And in 3D printing also, like if you are from 3D printing background, you know that in FDM and other process, like in triple F, fused filament fabrication process, we need some support structure. Here also, we can provide some kind of support material to print any complex structures, right? And it is truly so, because in many of our tissue, suppose you have to print this ear shape, it is a very complex structure. So that to support that the hanging geometry, we need to provide support. But the one criteria here is whatever the support material we use, that should be biocompatible with the thing, and also that should be water soluble, so that we can easily take out the material. The other thing also we can do, we can combine both this, like the FDM and the extrusion based bioprinting simultaneously, not together. Suppose the first, only the thermoplastic polymer that is printed in a layer by layer manner to get the template. On top of that, now we can print the hydrogel, so to get the 3D structure. Now the next printing technology is the laser assisted bioprinting. This is a little different from the other two. Here what we have some kind of ribbon like structures. Like here the ribbon like structure, the ribbon is consist of the very first layer is a glass or quartz, and the next layer is either it is an energy absorbing layer, or it is also maybe made up of mostly gold, and then the top below most layer is the Biowing, that is a cell laden hydrogel viscous material that is coated inside this thing. Now whenever the laser is on, what it will do, it can pass through the glass slides and it can hit the gold layer, and the gold will get heated up, so that it will get heated up and then it generates heat. So by this there will be Biowing, it is the aqueous based layer, so some bubble will be produced. and then but due to the bubble produced, that will dislodge some material from this thing, and that can be collected in the collector plate. Now, depending upon your CAD geometry or the CAD model, this laser will move on the top surface, creating different patterns, and then by this, droplets will be will be generated from different areas, and that can be printed. This is the thing, like the CAD model, you generate the CAD model, and then that CAD model is used for printing the structure. Here, this is the thing. Now, whenever the laser is falling, we are striking on the gold, is getting heated up, a bubble is produced, a droplet can be transferred. That's why this technology is also called laser-induced forward transfer, lift bioprinting. Here also there are various process parameters, like some process parameter related to laser, viscosity, surface tension, all these different process parameters are there. You can see in this video clearly how this process is actually done. You see here the laser, they are showing as a photon. Suppose the photons are passing through, striking the mirrors and getting deflected through the mirror, and that is through a lens that is being focused, and that is striking this ribbon. And whenever there's striking the ribbon, it is transferring energy, gold is heated up, there is a drop formation, a droplet is getting formed, and that is being printed. Like in a layer by layer manner, these droplets can be generated one after another, and that is being created, and that can be created for the fold structure. Now there is a recent development, because many a times if you are working with hydrogel, you know that with the Biowing, means the hydrogel like Biowing, it is very difficult to make 3D structures. Now there is a liquid. Recently there is a different technology where 3D printing can be done in a supporting bath. Supporting bath means where this bath is also made up of some hydrogel, some supporting gel is there. Inside that, we can actually basically immerse our needle, and then we can start extruding material there and there itself, so that this gel that supports the architecture, right? So then in this process, we can create any types of structure, any types of geometry. This is a video from the MIT Media Lab. You can see how exactly this process is done. Now suppose this is the gel bath, like the supporting bath. Now this one needle is immersed here, and then they start extruding the material within this gel. You see how the material is being extruded within this hydrogel. The gel is supporting the material, is holding the material to keep that on that place. And again, any kind of geometry that can be used. Now you see, and then that can be taken out and that can be used. This is a very good technology for printing any material. You see, in our levels we used to do that. Now this is the thing, when you try printing only the hydrogel, low viscous hydrogel, you're not able to make any structure. But the same thing when we use this inside a supporting bath, that you see, we can get very nice structures, right? This is a very good, very powerful technology by which we can develop any 3D structures using very low viscous material. And for bioprinting is true, because we cannot use very high viscous material, otherwise it will kill cells. So that's why we can create, now in this lab we can create a 3D structure. Like you see, this is like a cylindrical structure we have created in our laboratory. There are some properties the suspension bath should have, or the supporting bath should have. So there should be a critical yield stress, because unless we move, the gel should not flow, right? So otherwise they cannot support. Other thing is the self-healing, because when the needle moves away, it should be able to come back to its original position, the gel. So these two properties are very important when you use for this thing. Now recently there is a study from Israel. There, you know that probably you have, I don't know whether you heard it this or not, the human heart basically can be developed within this thing, within this supporting bath. I'll play also the sound so you can hear this thing.
In a major medical breakthrough, Tel Aviv University researchers have 3D printed the world's first complete heart using a patient's own cells and biological materials. Until this moment, scientists have successfully printed only simple tissues without blood vessels.
Technology that we've developed in the lab is based on a small biopsy that is taken from the fatty tissue of the patient. The cellular and the acellular material is separated, while the cells are reprogrammed to become stem cells and differentiated to cardiac and endothelial cells and other cells.
In the United States, heart disease is the leading cause of death among men and women, and heart transplantation is the only treatment available to patients with end-stage heart failure. Worldwide, only 4,000 transplants occur annually, and 20 people die every day waiting for a transplant. With a shortage of heart donors, the need to develop new ways of regenerating a diseased heart is crucial. The engineered heart matches the immunological, cellular, biochemical, and anatomical properties of the patient, which researchers hope will reduce rejection rates by the patient's bodies.
We look at CT images of patients, we look at the blood vessels, and we create matching tissue that, in addition to the match of the cells and the biomaterials in the patient, it also matches the anatomy of the patient.
The heart created by the researchers at Tel Aviv University is small, about the size of a rabbit's heart. It has the muscles, fibers, and blood vessels of a heart, but it is not yet able to pump independently.
So the next challenge would be to mature these hearts in the lab, interact better with each other to provide better electrical signal, so that the heart will have pumping ability. And then after we achieve that, we would like to go to small animals initially to implant these hearts in parallel to the hearts of these animals.
The technology is in its early stages, and human transplants are still a number of years off. But Professor Dvir hopes that within the next decade, organ printers will become a staple piece of equipment in hospitals, giving new meaning to the term "have a heart."
Yeah, so that is the bioprinting approach, or bioprinting in a supporting bath that was used by this Israel scientist to develop the thing. And that's a major breakthrough. But their next challenge, of course, as they mentioned, their next challenge is to mature this heart so that we can has this pumping ability. You know that there's all those different cardiac muscles, they can able to they contract, they can able to contract, but the synchronous contraction is absent still now. So that is the major challenge that we are working on. Now the major or the next vital ingredient of this, or the most important ingredient of bioprinting, is the Biowing. This is nothing but the cell-laden hydrogel, and it is also, it helps to provide a microenvironment to the encapsulated cells, so that the cells can function, so they can function and they can work in a good manner. Or it also actually it also helps the cell to viable, provides nutrition, provide necessary other conditions for this thing. There are certain key hydrogel properties that is required for this printing thing, like the biological point of view, it should be biocompatible, it should be biofunctional. From a rheological point of view, there should be sufficient viscosity, right? And the physical, there should be a proper yield stress, and also the gelatin mechanism that should be cytocompatible for this bio. Different types of biowings are there, starting from gelatin, alginate, collagen, hyaluronic acid, gelatin methacrylate, and all different types of biowing you can think of. In our laboratory, we specialize to develop a biowing starting from discarded human or animal tissues. So we collected the discarded human or animal tissues. That tissues is basically first it is desolarized. Desolarized is the process by which you can throw out all the cells from the material, so that only the material is remaining from the tissue, because that material is not immunogenic, the cells are immunogenic. So you have thrown out all the cells so that it can be used for tissue engineering purposes. Now that the next thing, we have developed the hydrogel from this desolarized tissue, and that hydrogel can be used for printing the cells can be loaded within this, and that can be used for printing this purpose. Now here, this hydrogel is used for printing three-dimensional structures. Now here, and then the cells are also loaded that allowed to mature, and that can be used for different purposes. Some more info about this work. Now this is the first. This is the native tissue. You can see the native tissues are collected, like cartilage, heart, or adipose tissues are collected. Then that is being desolarized, and then we have also characterized them by different ways. One thing such thing is the DNA quantification. Because if you do the DNA quantification, we come to know what is the level of what is the extent of tissue lysis, what the cells, because the DNA quantity should be very, very less. It should be less than 50 nanogram per mg of tissue. So that's how, so we reached there. And also we characterize the two different other major components of the extrusion matrix, that is collagen and GAG. So those things also are quantified. And the next thing we found that when we made the hydrogel out of this material, or this decellularized tissue, it is actually behaving like a temperature sensitive material. At 15 degrees centigrade it can flow, but at 37 degrees centigrade it can arrest the flow, and also it becomes gel. So then we found a very good way to prepare, and our radiological data also supports that. So then we printed the structure with this hydrogel. Like this is the printed structure with all the hydrogel. Also, as I already mentioned, with the hybrid printing concept, where the PCL, the thermoplastic polymer that is used to print the framework, and then we have printed the hydrogel in every gap between these things. So that can be so the hydrogel, the cells can be encapsulated within the hydrogel, and the PCL can be used as a template. So that's how we printed. So this is a video from our own work. The video from our own work, we can actually basically can see how this 3D printing can be done. First, the PCL layer is fabricated. PCL line by line, the PCL structure is printed. Here we have provided some gap between the lines, and then we are going to fill the hydrogel or print the hydrogel in every alternating gaps, every alternating gaps between the lines. And this hydrogel also contains cells. So the cells here we have shown two different types of hydrogels can be added. So two different, like blue and pink. So so basically we can position this hydrogel anywhere in the structure. Then we have done some in vitro evaluation. The first thing is the morphology of the structure. You see, this is the printed structure here. This black line is the PCL, and here this is the cells that is encapsulated or that is present within the hydrogel. These round things are the cells that is present. Also, we have seen the whole structure. You see, the hydrogel is present within the whole structure. So this hydrogel is very much stable till 14 days. Also, the cells are viable. This is viability data. This green dots are the live cells that is present within the structure. They are present everywhere. The structure is also stable long term, like till 14 days. The structure is stable. It doesn't disintegrate. Also, the cells are viable. You see here, the green dots, those are the cells, live cells that is present. Also, another thing is the cells can connect to each other, like three-dimensional interaction between the cells, which is very important for tissue building, and that is actually that we can see clearly that is there. The next thing also we characterize the tissue differently, like the gene and protein expression, because all different types of tissue, they have their own function. They produce particular types of protein, particular enzymes, hormones, all these things, different types of different organs, they present different function differently. So that's what we try to understand, whether our bioprinted tissue, they can express different genes or proteins or not. Like here, you see this our structure that produces different genes, and that is also compared with the collagen, because collagen is used as a standard material for bioprinting for various research groups. We compared that thing. We found that our material or our printed structure expresses more particular tissue, more tissue-specific genes than the other one. Also, in our immunohistochemistry result also show the same thing. You see, because as I said, the different tissues express different proteins. So here, the cartilage tissue, suppose they produce collagen 2. Like here, we see that in our structure, the collagen 2 is very much present. So that is the cells that is there. Accordingly, for other tissue also, different. We can see different types of proteins are expressed. So the tissue-specific genes and protein expression data tells us that the structure is, first thing, the structure is viable. By cell viability, you can understand. Then the structure is also functional, or this bioprinted tissue that is functional, because they are expressing different genes and proteins. Another thing we have done, as I said, the complex structure like ear, that can be printed within this hybrid printing concept, where basically the sacrificial material can be printed along with the structural materials in every layer. And when the whole printing is done, then basically we can dip the structure in water so that we can remove the sacrificial material. So we can get the three. So suppose this is a reverse pyramid structure. Here, these are the hanging geometry, right? So this hanging geometry can be very well printed within this approach. We have printed this kind of ear-like structure within this hybrid printing concept. Now, next part of my talk is to discuss a very other important application, that is in vitro models. You know that for drug screening, in vitro models are very much used, has to be used, because you know the drug discovery process is starting with the identification of various lead molecules, and then that gone through that, actually that passes through the cell culture studies, models, to then few of the molecules that passes through this thing, because other molecules they might have toxicity, they're not good. Then this few molecules then again passes through the small animal trial, then that passes through the big animal trial, like the monkeys and other things. Then that is being used for some voluntary patients. Then the next thing is that is also used for the treating patient. Now this process is very long. It needs huge amount of investment, and other things, there are ethical concerns because we are using animals for this thing, and many a times the response is also not well. So that's why there are recommendations from various funding agencies to minimize the animal use and to go forward with using human specific models, in vitro models. So that's why these human specific in vitro models are very powerful, because here many systematic studies can be done, repetitive studies can be done. These are high throughput, we can get the results very fast. Quantitative investigation can be done, different types of doses can be used, dosimetry can be studied. So all these different types of things can be done. So that's why in vitro tissue models are very used. In our laboratory, we are interested to develop in vitro liver model, because liver is it's the primary organ that does the metabolism in our body. So whatever we eat, that goes through the liver for metabolism, and that can be used, right? So whatever the drugs we consume by oral route, that is also passed through the liver, right? So the drug biotransformation, that is also done by the liver. So that's why liver is the first target of the drug. So the many of the drugs, if it is toxic, that can be toxic to the liver. So that's what we try to understand. And liver is a very structure of the liver is very beautiful. It's like a hexagonal. It consists of hexagonal units that those are called lobules, and the liver consists of multiple lobules, like the millions of lobules are connected to each other to the whole liver. So that's why this liver is a primary target of developing in vitro liver model. There is a company or the industry called Organovo in US. In 2014-15, they have developed a in vitro liver model by this extrusion-based bioprinting approaches, and then they have supplied that to the various agencies for taking the drugs. So this is a video from there. Here you will see how the liver model is developed, so that can be used for drug screening purposes.
Organovo creates functional human tissues with its unique bioprinting technology. Starting with cells from any source, Organovo's bioprinter precisely places a specially formulated bio ink, in this case, into a 96-well plate for drug discovery. The process results in a complex tissue with natural architecture. Disease can be induced, and disease progression can be studied in the native microenvironment. For medical research and drug discovery, potential treatments can be observed real time to measure safety, efficacy, and mechanism of action. 3D bioprinting can create more natural cell-cell interactions than traditional 2D methods, with better predictive value and higher clinical relevance. This revolutionary technology can speed safe and effective drug development, providing an entirely new way to study disease and advance medical research, and ultimately create a potential new source of tissues for surgery and transplant medicine. Organovo, changing the shape of medical research and practice.
Yes. So this is a video from their study, like where they have used this cell plus materials using this extrusion-based bioprinting approach to print the 3D structures. Now, this application of this kind of in vitro models can be various, like understanding the pathology, physiology, and pathophysiology, like suppose the structure is building, what pathology, physiology is going on, and then also we can also develop the pathophysiology. Suppose we use disease cells, then or induce some disease inside the cell, like liver fibrosis. Liver fibrosis is a disease that is like various patients, or many patients they're having this thing, and so to develop drugs of these things, we can develop this kind of liver fibrotic model, and then we can test different drugs whether this drug is actually effective on this to treat this thing or not, right? The next thing also, we can understand the morphogenesis of this process. Drug screening, as I already said, drug screening can be definitely done within this kind of models. So because this model, once it is developed, then various drugs can be applied or used on this thing, and then that can be studied whether the drug is having a toxic effect on the product or not, on this bioprinted tissue or not. Or suppose we reduce some disease, then we are using some particular drug, then whether this drug is effective to treat that particular disease or not, whether that is it is reversing this pathology or not. Like nowadays, you know that this the COVID-19, the corona, this disease is a pandemic, so many of the patients are affected. Now how to study this in vitro setting? How to study whether this how the virus is actually affecting the lung tissue, how it is spreading, and is there any drug that can treat the patients? Like, so how it can be done? Now suppose if we develop a lung model within this bioprinting, we developed a lung model, right? And this lung model can be used, then the virus can be inoculated on this lung model so that the virus can infect. Then we can develop a COVID-19 model, right? Then now, if somebody is developing drugs, then they can actually basically test their drugs on this infection model, and they can see whether this drug is actually effective to treat the disease, or that means whether the drug is effective to kill the virus or not. So that kind of in vitro disease model, in vitro infection model, that can be used, right? Or like suppose in case of this COVID, in vitro COVID-19 model can be used to develop, and that can be used to treat. You have to find out the new therapy that is suppose some people are developing, that can be find out to evaluate that can be used. Other thing is that we can also develop cancer models within this proper approach. Like the in vitro cancer models can very well develop, and that can be used for screening, anti-cancer drugs, or some novel therapies that can be used. So in vitro cancer models are developed out of this thing. We are also working in various on this lines, various projects are going on. And so now we have almost reached to the end of the talk, when I'd like to discuss a very important thing with all of you. Probably many of you have this question, like suppose everything is well, now suppose everything is going good, then when we'll get this artificial tissues or organs that can save patients? Now you know that every 30 patients, in every 30 seconds, one patient dies because of non-availability of artificial tissues or organs. So there is no donated organs available for that patient. So there are every 30 seconds one patient dies across the worldwide. So if we can develop artificial tissues or organs, then definitely it can be a boon, or it can be a helpful, beneficial for many of such patients who are awaiting for organ transplant. But there are few technological challenges, also few regulatory challenges in this area. Like till now, no bioprinted products have been put into clinical human trials yet. Like clinical trial is not going on. There are various reasons for this. Like there is, as I told, the technology is not that mature, so they can develop solid organs. Also, the regulatory body, they have to come up with their regulations of how they can actually approve this bioprinted product. So that is another challenge. But nevertheless, in the short term, there are like two-dimensional tissues or organs that can be developed, like hollow tubes, or tissues that can be developed. In mid-term, hollow organs can be developed, and long-term, solid organs. Like this is I have taken this from this website, like joint cartilage, vertebral disc, skin, peripheral nerve, meniscus tissue, that can be bioprinted soon. Like over the next 2 to 5 years, we can able to get these things. Like human trials can be started anytime, and then we can able to that can be used in the clinic. But like some organs like trachea, heart valves, anterior cruciate ligaments, these things, this may take little more time, like 5 to 10 years it may take to start the clinical trials, also to get into clinics. But the solid organs, if you're thinking like somebody like liver, kidney, all these things, it may take little longer. Maybe it may take some more than 10 years time to start clinical trial, and also to develop the tissue, and also it can be used so that patients can be died. Let me acknowledge many people, because this work is a collaboration with various scientists across the world. Also, like in my PhD students, many of the PhD students are working on these different aspects of this bioprinting, and thanks to them, because they were working hard in this area to develop various types of tissues organs. I'm fortunate to get funding from various funding agencies, because this work needs extensive funding. Yeah. So thanks to all of them. So definitely this is a very exciting research area we are working on, and we hope that definitely we can contribute significantly in this research area to develop various types of tissue organ, and we are working on various fonts. Like I don't know whether you have seen or not, recently, there's a news in media newspaper. One of our work is published, that is we are working on bioprinted of cornea along with this A.B. Prasad Eye Institute. So there we are very close to developing this bioprinted cornea. We have done some NPV studies and we found exciting results. Probably now we are awaiting to get approval, and so that we can start this limited clinical trial on this work. Though this, we are far away from developing artificial tissues or organs, but I am very much optimistic that definitely we will reach one day. We will reach reach a stage where we can be able to print the artificial tissues or organs, and that can save patients. So thank you very much, all of you, for listening, for attending this talk, for listening, and I will be very happy to answer if you have any questions. Also, if you can also write to me. If you have any questions, comments, concerns, if you have some novel ideas, specifically for the students, and also for few the faculty present here, if you want to work in that area, if you want to collaborate with us, we are very open to collaborate with you. And if you have any novel idea, you want to try that out, you are most welcome. We have facilities like we have bioprinters, we have other facilities. You want to work on that, you are most welcome. And also you can write to me, like my email ID is falguni@bme.isth.ac.in. You can note it down. You can write to me if you have any question, comments, concern, any novel idea. I'll be happy to hear that. And also I'll be happy to answer if you have any questions. Thank you very much, all of you. And also I should thank you, Cambridge University, Cambridge College of University, so for providing me the opportunity to deliver a talk here in this FDP. Thank you all. Thank you.